Okay, I don't need to introduce myself. We will save some time. I'm going to talk today about increasing Clotho for a longer health span and lifespan. And I want to ask the audience, who has heard about Clotho? Oh, quite a lot compared to other audiences that I've been speaking to in the past. Okay, so I guess this is, yes, just some disclosure. So the question is, I'm sure at this longevity meeting, would you like to live 10 or 20 additional healthy years? And I'm saying that it would be realistically soon if we could prevent the decline or increase the level of the Clotho protein in our bodies. Oops. What is Clotho? Clotho is named after Clotho, one of Zeus' 3 daughters, the goddess who spins the thread of life. And Zeus had 3 daughters. Clotho spins the thread of life, Lachesis measures it, and Atropis cuts it, indicating death. Clotho was discovered or published in 1997 by Makoto Kuroho, who discovered it by mistake in a mouse that showed... Oh, sorry, I didn't see that. Okay. Thank you very much. He produced a mouse that had a very short lifespan and showed many signs of human aging, such as osteoporosis, arteriosclerosis, cognitive decline, and that was really interesting to him. And he discovered this gene that caused those changes. Then he produced an overexpressing mouse that had more Clotho, and that mouse lived 30% longer. So instead of something is wrong with this slide, but the normal mice live 24 approximately months. The overexpressing mouse lived 30% longer, which means they lived around 3 years. So I wanted to just give a very short background how I arrived to work on Clotho. It started with a question that we asked in monkeys: what causes cognitive decline during aging? And why do 50% of people, monkeys or rats, age successfully while the rest have cognitive decline? What are the changes that occur in the brain with age? And can we arrest or reverse brain aging? And these questions were the subject of a very long program project at Boston University School of Medicine, and it was supported by the Aging Institute of NIH, and the number of our program was 00001, was the first project by the NIA, by the National Institute of Aging. And what we found was that, this is the point, yes. Here we see the cognitive index of monkeys from age very young to very old age. And at the young age, if we are under the number 2 of CII, it means that they have normal cognition. If they go higher, it means that they have poor cognition, they have cognitive decline. So during the aging, we see this cognitive decline, and the question was, what causes cognitive decline during aging? And we did some analysis of the gene expression in the aging and young brain and discovered this gene Clotho that we didn't know anything about it. We saw that the Clotho protein levels and gene expression was very downregulated as a function of age. So we decided to try to understand what Clotho does in the brain to cause this cognitive decline. And we found that Clotho extends lifespan in many species, not only in mice, in nematodes, naked mole rats, in zebrafish, in mice, and in humans. So it was interesting for us to start to study Clotho. In human populations, and this is a study of over 10,000 people, people who had more clotto lived longer. They had less all-cause mortality than people who, in red, who had less clotto. This is the lowest quartile of clotto levels. They lived a shorter life. And then we can make a correlation between decreased clotto serum levels and the onset of all age-related diseases, like we heard before. As clotto levels decrease, and we can see during at age 20, 40, 60, 80, so does an increase occur of diseases of aging, such as Alzheimer's, cancer, heart disease, and so on. And we can easily measure clotto in the blood with a very good ELISA, and we know that at the young age it's around 900, then it goes down to around 800. We know that below 600 picogram per milliliter, we are already in the danger zone, and we will need to increase clotto levels. Interestingly, children have very high levels of clotto, and so do highly trained athletes and superagers. People who live above 100, they have higher levels of clotto. So clotto boosting could prevent or treat many age-related diseases that affect all organs of the body. I won't go into detail here because I want to talk about our findings. We found that in the Petri dish, clotto is neuroprotective, antioxidative, and anti-inflammatory to brain cells. In mice, we showed that clotto overexpression improves the outcomes in models of Alzheimer's disease, multiple sclerosis, and ALS. Alzheimer's patients have reduced clotto in their CSF when compared to healthy controls, and because of a polymorphism in the clotto gene, which changes 2 amino acids, and it happens in 20% of the population, these people, they can think faster and have an IQ higher by 6 points. They have a bigger cortex and live longer. But this is not something we can choose. This is in our genes. But it only tells you the correlation between longevity and health and cognition with clotto. So when we do measure the distribution of clotto serum levels versus all cause of mortality, we can see that if we are between 200 and 1400 picogram per milliliter, this is the distribution of the entire population. But actually the optimal range will be between 1400 and 2200. Then this is what we would like to achieve. But as you can see, the vast majority of the population is outside of the optimal range. So our goal is to shift clotto serum levels to the optimal range. And we are proposing to do this with clotto mRNA. And I'm showing here a picture from the X Prize HealthSpan Solutions. Maybe you heard about this prize. And that at the end, $101 million will be given to a group that can prolong life by, I forgot, 10 years. Peter, is it 10 or 20 years? I forgot, something like that. So our group, together with Peter Sramek, that will speak tomorrow, are 2 groups that out of 600 applications, they ended up with 40 teams that were chosen, and 2 of them work on clotto. So it looks like you can identify here Nir Barzilai and some other people that you may know that work on longevity. So our lab did research on clotto for the past 20 years in academia and industry. We showed different modalities of clotto. We worked on small molecules for clotto upregulation, different molecular biology methods. We used gene therapy with AAV and also clotto mRNA. We used animal models for some of the diseases like Alzheimer's, MS, ALS, Duchenne muscular dystrophy, cancer, kidney, and weight loss. We tried to see how Cloto can affect all these conditions that are, of course, age-related. Okay, I don't have to go into the mRNA LNP vaccine for COVID that all of us heard, but this is when I got the idea and maybe an aha moment in 2020 to try to use mRNA not for preparing a vaccine, but for protein replacement therapy, which is kind of the opposite of a vaccine, because when we inject an antigen, we want to make antibodies against that antigen. Here, we inject the protein, we don't want to make antibodies against the protein. The opposite, we want this protein to increase its levels in order to stay there and have an effect. So the way it works, we prepare Cloto mRNA, we encapsulate it in lipid nanoparticles, and via an IV injection, we know that such particles go to the liver. In the liver, the hepatocytes take the Cloto mRNA and produce the Cloto protein and secrete it into the circulation. And this is what we need. We need Cloto to circulate. Cloto is a hormone. It goes everywhere in the body and it affects every organ in the body. The reason mRNA replacement therapy is beneficial, it goes into the cell cytoplasm, it is transient, so we can control the levels of the protein expression. Unlike AAV, we can adjust dose and frequency of administration. We can administer multiple times without immune barriers, no risk of insertional mutagenesis, and lower long risks, such as long-term risks, such as cancer. It's suitable when proteins are difficult to make and deliver as drugs, and many companies and research groups try to make the Cloto protein itself as a drug, but they failed because the protein is highly modified, post-translationally modified. The mRNA lets cells produce complex, properly folded, and post-translationally modified proteins in situ. So if we give them to a person, then they will make exactly the protein that is naturally made, unlike a recombinant protein. The mRNAs can be modified to evade innate immune recognition, reducing side effects. LNPs and other delivery vehicles can shield mRNA until they reach their target tissue, and no need for costly purification of protein therapeutics from mammalian cells. And the mRNA manufacturing is cell-free, scalable, and consistent. So this is an example of our proprietary Cloto mRNA construct that yields more protein at a given dose in vitro. And in blue, we show that we can make, with the proprietary sequence, a protein that is 5 times the level of the native protein or the native sequence of the RNA that we prepared. And as a result of that, we can use lower doses and reduce the side effects if there are any. And of course, we can reduce the cost. So these are some examples in vivo and in vitro. We can see that the mRNA, we can, here what I show is the protein measured by ELISA. There is a very good ELISA for the human protein. And in cell culture, we can get 5 times the protein compared to the native sequence. And in mice in vivo, we can get twice as much of the protein by injecting the mRNA. Here again, I'm showing that we can get 2 times more in mice. And in some experimental models, for example, on the right, here we show that while we gave 6 times injections of Cloto mRNA, we can see a reduction in weight. So it can be used also for weight loss without any negative side effects. The moment we stop the delivery, then the mice regain the weight. Cloto is also tumor suppressor. We tried a model of pancreatic cancer, and we can see on the left, the upper panel, the mice with the big red tumors. The lower panel, the mice that got Cloto, they have very small tumors. And on the right, we measure the levels of the size of the tumor volume, as a function of the levels of the Cloto in the blood. The more Cloto we had, the smaller the tumor, like the very low blue line. You can see the 2,000 picogram per milligram, which will be a normal level of Cloto in the blood. Then we can see no tumor growth at all. And that was with AAV. Then we tried with mRNA. We got similar results, but with only one dosage. If we use more, probably we'll get also lower tumor volume. So finally, under the named patient program in the EU, which is allowed, patients were treated with our protein replacement therapy. And we can see here the levels of the Cloto protein in a 50-year-old male. And this was done at the Healthy Longevity Clinic. And Peter Sramek will talk about it tomorrow. And you can see this person started at 1 picogram per milliliter. And after maybe 24 hours, he already reached the 3,000 picogram per milliliter, which is a amount of really Olympic athletes. And it took some time, and then it started to decrease as a function of time. But even after 2 weeks, already still it was above the background. So it can work in people. So in conclusion, we have shown the therapeutic effect of Cloto in animal models for cancer, weight loss, kidney disease, Alzheimer's, MS, and ALS. And given these results, it is highly likely that Cloto supplementation will have a beneficial and perhaps even preventive effect against these human conditions, leading to improved health span and lifespan. Because I don't have much time, and maybe it will be nice to have some questions, I will just want to thank you a lot, and to thank all our collaborators from Advantage and Clotea in Austria, Art Bioscience in Israel, Science in Action in Israel, who collaborated with us, and the Healthy Longevity Clinic in the Czech Republic. Thank you. Yes.
Thank you. Is there any study looking on the homeostasis of Cloto in the serum, namely the level of degradation versus the expression of translation of the mRNA? Maybe inhibiting the degradation is also a way to increase the levels of Cloto.
You are absolutely right. Inhibiting the degradation will help. We just don't know which enzymes in the blood are responsible for the degradation. And also, it will be much less specific, because I assume that the enzyme that will degrade this protein in the blood will also degrade other proteins. So you are right, degradation and production have to...
Anybody looked on the changes? Is it due to degradation or less expression?
Of the native... So in mice, we didn't see less expression of the endogenous one. In mice. In humans, we haven't tried.
This is a fantastic talk, really. So it's here.
I'm going
to ask a question. So it seems like Cloto can do so many things, and can have so many indications. I wonder what indication you will target first. This is for longevity, or for the weight reduce, or for the cancer. Seems like the cancer it's really very remarkable results. Now, with regard to my second question relate to the clinical trials. You say 1 milligram or whatever it was dosed to the patient, to one person. So how did you do this experiment? Did you do under IND or just like RT related, and where is it?
Okay, the case study that I showed. Okay, so maybe Peter will talk about this tomorrow. He will go more into detail. In Europe, it is allowed to have, if the physician agrees and the patient agrees, then it is done legally. So in the United States, I don't think we can do that yet. But we are, of course, in Israel. I don't know the rules here. No, we had 7. Yeah, but this is all very legally done, of course. And the first question about indication, this is a very important question. And we are debating still, because longevity is not an indication. Longevity is not considered a disease. And I know that people in the longevity crowd are trying to show that if we can increase health span and longevity, it is preventing diseases. So this is something really very positive. We can do that. But still, you cannot write a grant for longevity. For example, you are not going, as far as I know, it is not an indication. Exactly. Well, you cannot wait 50 years to see if that person lived longer, I cannot. Maybe you can. So in any case, we are thinking about metabolic syndrome. And for cancer, there is a very fantastic group at Ichilov, Ido Wolf and Tammy Rubinek, who work on cancer and Clotho, and I leave this for them. We are also thinking about kidney disease, because I didn't mention that kidney and brain are the most major organs that produce Clotho, and kidney diseases are also on our list. As I said, metabolic syndrome. We are also looking at some orphan diseases, one of them being ALS. But to decide which one is first, we are still working on it. We have to go through clinical trials, phase 1 first. And we still have a little time to decide. Yeah. Yeah, correct.
Thank you for a great talk, very interesting.
Thank you.
Findings. I'm just curious to know, many slides that you show, the lifetime of the treatment is decreasing very steeply after 72 hours and or a couple of days from the treatment. So what is your vision of the company, of Clotho? How to administer this to the humans? Because
very important
question. Seems to be like multiple injections or
yes, very good
supplementation.
Yeah. Okay, so you are going to see tomorrow in Peter's talk that although the Clotho levels decline, let's say after a week or so, but the effects of Clotho persist, and they persist for a long time. Clotho starts, I didn't go into the details of all the pathways, biochemical pathways that Clotho starts. There are a number of them. And based on these results that you'll see tomorrow, we propose to give this injection once a month. Because if we give it once a month, the effects of Clotho will stay there, even if Clotho is reduced. It has long-term effects. Thank you very much. I don't want to take any time from the...